Hi, my name is Boris Rosin and I'm an MD-PhD student at the Hebrew University Medical School. The MD-PhD program is a program that combines studies towards an MD degree, medical doctor degree, together with studies towards a PhD degree in one of the basic medical sciences, in my case neuroscience. My PhD was done at IMRIC, the Institute for Medical Research Israel, Canada, and ELSEC, the Edmund and Lily Safra Center for Brain Science, and I work at the laboratory of Professor Bergman. Professor Bergman is one of the world's leaders in the research of Parkinson's disease, and thus my PhD deals with Parkinson's disease. To those of you that are not completely familiar with Parkinson's disease, it is one of the most prevalent neurodegenerative disorders of the human brain, and it consists mainly of three motor symptoms. The first one is the appearance of akinesia or bradykinesia, the lack or the poverty of movement, respectively. The second one is muscle rigidity, and the third one is the appearance of resting tremor, which is perhaps the most famous symptom of the disease. It is a 4 to 7 hertz resting tremor. So my work was done in two main parts. The first part was an attempt to point out the neuronal correlates of the Parkinsonian motor symptoms which we have just described. Differently put, to point out the exact neuronal discharge patterns that are responsible for the appearance of the Parkinsonian motor symptoms. This was very important for the second part of the work, which was an attempt to improve an existing treatment of Parkinson's disease, which is deep brain stimulation, or DBS. So, how DBS is performed today? DBS consists of an implantation of an electrode in a deep area of the brain, the basal ganglia, an area that is involved in the appearance of Parkinson's disease, and the delivery of a high-frequency, low-current stimulus to the area. This results in a drastic alleviation of the Parkinsonian motor symptoms and a great improvement in the quality of life of Parkinsonian patients. What we've tried to do is to improve the treatment in such a manner that the stimulation is not delivered all the time constantly as it is today, but rather is delivered in order to disrupt ongoing pathological neuronal discharge patterns. This type of stimulation, or this type of deep brain stimulation, we called closed loop deep brain stimulation, because in essence it closes a feedback loop between the brain activity and the delivered stimulus. We have shown that this stimulation in the animal model that we used for the study, which is the MPTP-treated non-human primates, was in fact superior as compared to standard DBS in alleviating Parkinsonian motor symptoms, which of course is very exciting to us, and for more than one reason. First of all, naturally, which this is a promise for a new treatment for Parkinson's disease, which is very exciting. And the second is the fact that such an approach could perhaps be useful in other brain disorders in which we can point out pathological neuronal discharge patterns. Such disorders include, for instance, schizophrenia, which has similar patterns of discharge to those appearing in Parkinson's disease. These results were recently published, and as I said, we're very excited to have them. And I would like to thank Professor Bergman for letting me be a part of the research and a part of his lab. It has been a great experience for me. And that's it. I would like to thank you for listening to this vlog. Thank you.